Good evening. I'm Frank Snyder, the dean of the Keese School. And do any of you have cell phones? Please don't make them beep during this talk. So we need food to sustain life. Beer is food, but it's more than food. Our speaker, Robert Bryant, or Rusty, will describe some remarkable byproducts coming from beer production. The title of his talk is Beer, Burps, and Greenhouse Gases. This is going to be fun, isn't it? Rusty received a PhD in biochemistry from Florida State and joined biochemistry faculty at George Washington University Medical School. He did research in many areas, including the discovery of natural taste and flavor modifiers. Boy, what a guy. Rusty and his wife, Mary, moved to Asheville, North Carolina 12 years ago when he started the Asheville Sustainability Research, LLC. He started that to look for hidden values of spent craft brewer's yeast. Anyone ever brewed yeast, brewed beer? Yeah, you need yeast, don't you? Yeah. During this quest, he worked with scientists and students at the local universities and the Highland Brewing Company, I wonder why, and Warren Wilson College, where he served as an adjunct in chemistry. Rusty and Mary are residents of Edwards Fisher. They have a raspberry garden prod. I bet you've seen them out there in the garden. And Rusty plays pickleball. Let's welcome Rusty Bryant.
Hello, can you hear me? Thank you, Frank. It's a pleasure to be here and talk about our project, which started about 10 years ago in Asheville. And yes, so we're going to talk about beer. The burps are not after drinking the beer, it's from the cows. And it involves methane primarily. So one of the main things that I'm going to talk about is the yeast. So it's really disposed of with the smaller breweries, but what we've done is show that it's got some real value to it. Now, there'll be time for questions at the end, but we've just started a small company called Time Inc., and we're trying to, at some point, sell this stuff to large feed companies. So we've got some proprietary stuff, but it's quite exciting. So we started, we spent 30 years in New Jersey. We lived near Patterson, near the Great Falls of the Passaic. I spent a lot of time in the labs. Actually, my first paper came from Syracuse College of Forestry in '68, studying trees, as you might guess, chemistry of aspen trees. And then in 2011, we moved to Asheville, way in the far corner of North Carolina in the Blue Ridge Mountains. And we were there partly to help our son, who converted a chicken hatchery into art studios. That was a lot of fun for the first couple of years. So Asheville, when we moved there, was really booming in terms of craft breweries. So this is Highland Brewing, and they're large tanks, 2,000-gallon tanks. They've got a whole bunch of them. They started really in the basement of a local bar in Asheville, but now are a very large and older craft brewery in Asheville. But they've been joined by a couple of other bigger ones, Sierra Nevada, which is out near the Asheville airport, and then New Belgium, which is right across from our son's property, and I'll show another picture of that in sort of a sad situation. But the point is, if I can get my clicker to work, there were 57 breweries in Asheville, and in total they generated 2,000 tons of yeast a year. And so it's a big waste disposal problem for the smaller craft breweries. And What we believe is that it's a lost resource. There's a lot of good stuff in it, so you should not just treat it as fertilizer or send it down the drain. So when I first joined, we came to Asheville, I was sort of interested in kind of continuing a little bit of pharmaceutical research, and I was told by one of the biotech guys, Jonathan Lowry, that no, we're not doing any drug discovery in Asheville. That's done over in Raleigh, and NC State and all of that. But we have all this leftover yeast, and what are we going to do with it? So I got interested in that. And but I have to touch base that one of the reasons that these breweries sought out Asheville was that we just had wonderful water. So this is Triple Falls near Brevard. You may have recognized this picture. It's been in Hunger Games and Last of the Mohicans. And the interesting thing is that the water in Western Carolina, it comes over granite rock, so it doesn't pick up calcium and some of the other things that can be a problem for brewers. So speaking of brewing, it's one of our older technologies. It goes back 5,000 years, but it's, let me see here. It's a rather simple process. You take grain, usually barley, but they use others, and water, and you steep it like a tea. And then you take the juice, if you will, add hops to it or some other flavorant, boil it to kind of sterilize the solution. The Germans called it wort, and we'll mention that in a minute. And then you add the yeast, the wonderful yeast that does its magic, and after a few weeks, you'll get beer. So, now, if you've gotten into brewing, unless you do home brewing like Norm, there are a lot of crazy terms, some of which go back to the Germans. But the barley they use has been sprouted usually, and then it has this process called mashing, which is really just making a tea out of it. And then it generates this material called wort, which has the sugars from the barley along with some proteins. And then this gets boiled, and during that boiling process, a lot of the proteins and the polyphenols that have a lot of antioxidant properties that have come out of the barley, they end up precipitating, much like egg white when you heat it up. It doesn't take too much heat to precipitate it. And so this material is called trub, and it's another one of these things that the brewers want to get rid of. If you leave it in the beer, it makes the beer extra sour, so it's something that brewers want to get rid of. Then, of course, there's the hops. We'll come back to that. And then there's the term spent yeast, which sounds to me like, well, it doesn't have any more use. It's been spent, But no, it's really, what it means is surplus yeast. And actually, some of it gets used to start the next batch of brewing. So it's really, if you do any work with breads, it's a starter. Then, of course, there's the IPA, which stands for Indian Pale Ale. So you see a lot of IPAs for sale. And that goes back to the fact that if you add hops to beer, the British found this out, they could take their beer and sail it all the way to India, and it would still be good. It wouldn't have spoiled. And then finally, the heart of the hops are the lupin glands, and I'm going to show some pictures in a second. Okay, so here's a hop yard. It's out in Yakima, Washington. And so these things, if someone were standing here, these things are about 3 people high. They're about 20 feet high. And if you get up close, these are the hop cones. They're about 1 inch long. They're like mini pine cones. And then the heart of it are these, inside of each of these cones, are these glands called lupin glands, where the key components, the humulones and the lupulones, are made. And these are termed the bitter acids, or sometimes I'll say hop acids or alpha and beta acids. So craft brewing has really had a big growth in the past few years. And this data is really almost 10 years old, but there were over 5,000 breweries in the US, and they were making 25 million barrels of beer. A barrel is 31 gallons, once you get into that. And that ends up being about 10% of the total beer consumption. So it's by far not the largest component. But interestingly, craft brewers use a lot more hops. So 1.5 pounds of hops per barrel, which is 4 times more than Budweiser or Heineken. And craft brewers are really good for the community. Many of them open in old industrial areas, and I can tell you, working at Highland Brewing, they're a very good employer, and people are quite happy. Now, there are concerns, however, and they use a lot of water to make beer and keep the place clean. Being sterile is very important to breweries so that the beer doesn't spoil. And as a result, they generate a lot of waste. So that's something they want to deal with, but it just has it's a challenge. So, and a lot of the waste streams that we'll talk about are just burdensome for the breweries. They have to be very careful. If for some reason they end up releasing a large slug of yeast going down the drain, it can cause problems for the water treatment plants. And finally, the breweries generally have to pay for hauling this waste away. So that's, their margins are not big to begin with, so those expenses can be a problem. So, worldwide sustainability is something everybody's interested in: universities, manufacturing, governments, nonprofits like Asbury. The problem is there's a challenge. So, to utilize waste streams, you really have to show that it has a value, typically. And so that's what we've been doing at Highland and Nashville Sustainability. We've been working over the past 10 years to find the value of these leftover brewing products, particularly the trub, the hops, and the yeast. And we've actually coined the phrase time to encapsulate that material. So, trying not to fall off the edge here. So this is a kind of it's a bit detailed, but the flow chart. And the first thing I want to focus on is the spent grain, because people have been getting rid of or have found a home for spent grain since sort of the beginning of brewing. So again, the main parts, the 4 components as the Germans defined it for making beer. You've got barley, you've got water, you've got hops, and then you've got the wonderful yeast. These are the budding yeast cells. So once you have made the wort and steeped or mashed the barley, you get a lot of spent grain. And so it looks sort of like oatmeal, and animals love it. So here's some goats. I was talking with Sierra Nevada, and they had a regular arrangement from their Colorado site out to beef farms in Colorado. So getting rid of the grain is not a problem. But the surplus yeast is. So looking at where the yeast comes out, it comes out of the fermenter. So here's a fermenter. So we've put in the wort with the sugars and all of that. The yeast grows, multiplies many times, and then it all settles to the bottom. And once the beer is ready, you can drain it out. And so it ends up being about 1 to 2% of the volume of the tank. So it's fairly significant when you really think about it. And interestingly, it uses about 60 to 70% of the sugars that were released from mashing the barley. So it contains a lot of the nutrients that were, steeped out of the barley. And so it looks kind of like a thick pea soup. And if you're at Budweiser, they have a very well worked out waste stream, and it's made into a powder, and it's used for pet food. But for those of you that are really ambitious in your taste, you can find jars of Marmite, at least down in Crawford, and Marmite is made out of spent yeast, and it's the hydrolysate of the yeast. And I will say this: it's an acquired taste. The smaller breweries, the microbreweries, a lot of it goes down to through the main sewer lines, and they pay a price for that. So when we got to Asheville, we said, well, is there a food opportunity here? And I rented a lab in one of the natural products plants houses in Asheville, and we could spin it down, and it sort of looked like peanut butter. And I said, gee, we got to be able to cook with that. And I talked with Mary, and we thought, well, let's try it and see what we can make. So we ended up making brownies, and there's the yeast, about a quarter cup. Of course, it's got a lot of, we touted its health value. But as with many brownies, a lot of butter, a lot of sugar, then of course a lot of cocoa. Oh, and we had other goodies, but honestly, there's one of our batches, but it took 40 variations of our what we call brew bites before we had something that we thought tasted okay. And one of the secrets Mary found, actually going online and looking at some patents, peaches or other stone fruits, she found a Japanese patent, blunted the bitterness of some of the hops. So off we went, and we spent a while in Asheville at farmers markets and so on, selling our brew bites. Mary also kept the books. And what we found was we lost money. Anyone that's tried a small food business, it's got challenges. So I decided to go back to the lab where I knew more about what I was doing. So I went to App State, which is about 2 hours north of Asheville, way up in the mountains. And I worked with a very brilliant fermentation chemist. He actually focused on wine, Seth Cohen. And we wanted to figure out what was in this craft yeast. Had an interesting flavor, and Seth had, we needed was a liquid chromatograph, mass spectrometer, big words. It was about $200,000, one of the expensive pieces of equipment you need. And it would give you a profile of all these peaks of different compounds, and then the mass spectrometer would give you an idea how big it was, how many atoms were in it, et cetera, how it broke apart. Essentially, it gave you a fingerprint. That's all we needed to know. It gave us a fingerprint. And frankly, this sort of equipment is used in the forensics labs, and these mass spectra are valid in court. So they're seen as very powerful. So this particular peak here, the tallest one, is this compound, humulone, and it's very interesting. Another fact is that during the boil, that's the little fire here, it rearranges to isohumulone, and two things happen. It gets more bitter, so it gives you the IBUs you want, but it loses its antibiotic potency. So I'm going to get a sip of water. So what we wanted to do was compare the craft yeast versus, I'd gotten some samples from some folks in Wisconsin, samples of Budweiser yeast, Heineken yeast, and Corona. And when we analyzed all these yeasts, we found that the amounts in the craft yeast were much higher, 10 times higher in some cases, which was really, it was very exciting. We ended up giving a talk on that at a brewing conference. So that was really quite surprising. So we then decided, about that time, the folks at Highland Brewing, where we were getting the yeast slurry, said, you know, there's some other stuff that might be useful, and that was the trube. So here's the excess yeast that comes out, and then the trube comes out from the boil. This is the stuff that precipitates, kind of like the egg white. And what we decided to do was to combine the trube and the yeast, and there are also extra hops in there, and that's where we get the name thyme. And what we found was that the thyme had much higher level of these hop acids, the humulones and the lupulones, than the yeast. So these red bars are the thyme values, and then the blue are the yeast. So you can see that this is our humulone, and you can see how much more of it is in the thyme. And then this is the lupulone, and that's interesting because this is a much stronger antibiotic, and we'll get into that in a minute. So it looked like the thyme really, by combining everything, these things that are thrown away, gave us something rather interesting. So this is actually a picture of some of the thyme powder. We figured out how to dry it. And I've got some samples of hops and thyme, if anyone wants to see it at the end. So another aspect of the work up at App State that was quite surprising was we found that the hop acids, or the humulones and the lupulones, actually stayed in the yeast, and there wasn't so much in the beer. There was enough in the beer that gave you the IBUs and the bitter taste and other tastes that people like. But by far, most of the hop acids were in the yeast. And kind of an illustration, this is stuff that came out of a fermenter. It was really almost ready to bottle, but not quite. But so on the top is the beer, so to speak, and this is the yeast phase. So by far, most of the hop acids were in the yeast, and only a small amount in the beer. There was still enough to, give it flavor and all of that, but it's just amazing. These compounds are essentially very greasy, and they like to stay in tissues, if you will. So once we found out that we had these interesting hop compounds, the humulones and the lupulones, I started to look, what have people found with them? Because they've been around a long time. And so we know that the pure compounds can increase bone cell growth, block decalcification. There's some tests in test tubes. These are all test tubes, not people. But it blocks the growth of tumor cells. But most importantly, for what we were interested in, they had antibiotic activity. So they block the growth of gram-positives, and when they put lupulone in the water of chickens, it cut the number of some of these pathogens. So our question was, could craft yeast actually have antibiotic effects on its own? So I went to, instead of going north to Boone, I went down to Cullowhee, North Carolina, to Western Carolina, and found a microbiologist there, and we started doing some antibiotic activity measurements. So the way you do that, you take a Petri dish. This is very old technology. If you or your children or your grandchildren have cultures done, they often will look to see which antibiotics are really going to zap the bug that's bothering you. So you seed it with a few thousand bacteria or a throat swab or whatever, and then you incubate it overnight, and you get a thick lawn of bacteria. And if you'd put a little disc with some penicillin in it, what you'd see is the next day there'd be a halo around that. The penicillin was leaching out or diffusing out, and wherever it was enough to block the growth of this. This was probably an E. coli. So here's I'm sorry it's very complicated, but again, it's a Petri dish. And we had seeded it with a lawn of, this time it was Bacillus subtilis. And if we put a paper disc down with an extract from thyme, we saw these nice halos. And if you had less on the disc, you got less of a halo. So it's very clear that it had nice antibiotic activity. So the question was, where could the antibiotic activity of thyme be useful? So that's when we decided to look at cows. And so this is the rumen. Okay, it's the first stomach. The three other stomachs here, but we won't go into that. And then here's the going out the exit one way, going out the exit the other way. And it turns out that this rumen is about 25 gallons, and it has over 100 trillion bacteria, or I should say microorganisms, because you've got bacteria, you've got yeast, you've got viruses, and so on. And so 95% of the methane, it makes a lot of methane, and it uses this pathway. It's quite simple, actually, on paper anyways. You take CO2 and you add hydrogen to it, hydrogen gas, and the metabolism in the rumen generates a lot of hydrogen, and actually it uses this method to get rid of the hydrogen, because hydrogen can interfere with the digestion of the animal. So the methanogens, another organism, adds hydrogen to the CO2 to make methane, and then you have water on the side. And as a result, you get 125 gallons of gas, of pure methane gas coming out, and that's about about a pound of methane. And in fact, they burp it. So that's where the burps come from. So this is a graph where they're measuring both CO2, they have infrared detectors to measure both CO2 and methane. So here's the CO2, and he's just sort of breathing, all these little blips in there. Here's one cow. But here's the methane. So you don't see anything until it burps. Here's a methane burp. So over this minute or so, they've got three methane burps. So you see, these are four different cows, and they all have sort of a similar pattern. So methane from the cows is, over the past 20 years or so, we've realized that it's really an environmental problem. So in terms of total greenhouse emissions, greenhouse gas emissions, agriculture is about 9% of it. And of that, 9%, fermentation or rumen fermentation, where you're generating methane, is about 27% of that. So it's 2 or 3% of the greenhouse gases are coming from cows. So it's not the hugest amount, but actually it looks like that's about the amount of methane that comes from all of the petroleum industry. So cows are a significant contributor. So as a result, we all know there's been global warming. And this is surface sea level temperatures from the American Meteorological Society and from NOAA, showing the increase just in the surface water of the Gulf of Mexico. And as a result, we've had a couple of very serious hurricanes that have gone from the Gulf up into the south. And this was one that went up into Asheville, and then this is the result. The French Broad River was the highest it's ever been. When we moved in 2011 to Asheville, I'd seen levels of the 1916 flood, which caused huge devastations. But this was like 5 feet higher than that. So this is the New Belgium brewery that I was talking about. And the water's down, obviously, but they're still not making beer yet. And they're still cleaning up. So just to give you an idea how much water, there was an estimate on PBS that the hurricane and the one before it, there was a big storm before that, also coming off the Gulf. And together they put out 40 trillion gallons. So that's sort of hard to get your head around, except it's about 17 years flow of the Potomac River. So if you walk along the Potomac, there's a lot of water there. But this hurricane took the amount that took 17 years. I mean, it's fairly simple. It's like the flow rate is 10,000, normal flow rate of the Potomac, not at flood stage, but it's about 10,000 cubic feet a second. So that seems like a big number, but 40 trillion is a big number. So we were wondering, could feeding cows time these yeast products reduce methane emissions and help with global warming? So we had a very ambitious, skillful student at Warren Wilson, Virginia Kukulski. She's now a senior scientist at USDA in Madison, Wisconsin. And she did studies to show that yeast suppressed methane synthesis in test tubes by about 30%. And then we had some other students later who came along. And so Virginia was looking just at the yeast the stuff that comes from the bottom of the fermenter. We didn't have time at that point. And when we had time, other students came along, and we found that putting time in, about 0.5%, not a large amount, suppressed the methane production in the test tube, in the bottle, by about 80%. So the real question is, what about real cows, live cows, like, burping out methane every few minutes or every few seconds? So at this point, it's like, what's the big question in science? Where's the next grant coming from? So we are trying to find research funds. It takes about $100,000 to do these studies. Now, you could actually measure methane coming out of an animal pretty easily with kind of like the old radar gun that they would show, with the policeman, watching your car coming down the road. But you can see it coming out, but it's not a very quantitative way. So there was a place called Sealock up in Wisconsin that developed a head box. So this is it's like a mini closet, and it takes a while to get the cow accustomed to it. They're a bit nervous, but they put molasses in there, and eventually the cow will go in and it will stay in there, and they'll get enough good data to show that you have lowered methane. So there are a number, besides our approach using the natural brewing wastes, other approaches that have been going on and have frankly had more news. One is called Bovaer. It comes from a Swiss company, DSM, and it uses a compound called 3-nop, or 3-nitroxypropionic acid, and it drops methane about 30–40%, which is pretty good. The problem is this nitrosopropionic acid is very irritating, and it's not something farmers can easily use. So how well it's going to be used is not clear yet, and people are just starting to try it out. But it does work. Now, there's been a lot of news lately about seaweed. So there's an Arabidopsis species that is loaded with, this is bromoform. So it's the bromine sister of chloroform. And so it does, both of these compounds block the formation of converting CO2 to methane. Kind of there's a metabolic pathway. The problem with bromoform is that there's some studies that suggest it could be a human carcinogen, and the problem is that it could build up in the milk or the fat. So there's a lot of interest in it, but it's not clear whether it's going to be approved or not. Now, just a few weeks ago, there was an interesting story in Washington Post about a group that is using molecular biology to try to engineer microbes that you could give to a cow, and they would proliferate in the stomach and block methane. So they're using this CRISPR technology, but it's very early, and it's going to be a while before something like this might work. The upside of this would be you might be able to give each animal a probiotic, if you will, and to turn off the methane, but we're not there yet. So, having worked in several labs, I just think we have got to defend science and its funding. So it's very nice to join a group of us from Asbury at the NIH rally last month. And actually, I heard today that one of the local churches went to the NIH, the very same metro stop, and sang thank you songs, if you will, to NIH people coming out of the metro. So in summary, I think we've captured and demonstrated the value of some craft brewing co-products that have essentially been tossed away before to improve sustainability. So in a nutshell, we've taken the the trub and the hops that come from during the boil, and then the yeast, and we put it in this material called thyme. So it's been approved in North Carolina as a feed supplement. We've given it to chickens. We had several studies at Virginia Tech. We're very excited that it could help with methane mitigation, but we need a POC, a proof of concept, and that's where the money comes from. But we've had some interesting data recently to show that the thyme increases feed efficiency in cattle about 10%, and very recently we've had some work with pigs at North Carolina State that seems to indicate that it improves the health and the growth of weanling pigs. So we're quite excited about that. So this was we had a lot of support. Of course, we've had some pretty good grant funding, USDA. Most recently, NC Collaboratory, which is a state-funded granting agency, gave us money for the study at North Carolina's NC State. But I really have to give a lot of thanks to my colleagues at Warren Wilson. So Langdon Martin, professor of chemistry, was chair at Warren Wilson. Kim Borges was the head of microbiology. They were both very helpful. And then I've had the pleasure of working with about 10 or so young students at Warren Wilson. And Isaac he's working there now, and he did some work to show that thyme had a lot of interesting antioxidant activity. So, if you're talking about naturals, green tea, that sort of thing, antioxidant activity is a very good thing. So, and then I have to thank my colleagues at Highland Brewing. Oscar Wong was a retired nuclear engineer who was working in Charlotte and then moved to Asheville and decided to go into brewing. And he started Highland back in 1990. Then his daughter, Leah, became the president, and then her husband, Brock, is the vice president. So they've been really pushing sustainability. I had a picture. They were on Good Morning America last week because Highland, they were high and dry. So unlike some breweries on the riverside, Highland did not get flooded. In fact, they became a center of kind of recovery operations. So people were thanking him for that. And finally, I have to give most support and thanks to Mary, who's been a guiding light. So thank you.
Take questions?
Yes.
Please raise your hand, and the ushers will bring a mic to you. Back in the corner.
You mentioned spent grain, and I worked in Seattle at a bakery that was using the spent grain to add to their bread to make a more fortified loaf. And I'm wondering, with the yeast, could the yeast actually be used in the baking process, or is this more of a nutritional yeast?
Well, I'd say you're right. It's like nutritional yeast, except if you put a glob of nutritional yeast on one of those Petri dishes, you won't see any halos. It doesn't have the antibiotic properties. But, and I think in our brew bites, we couldn't go too much more than 3% of the yeast in there, or it got too bitter. So, yeah, nutritional yeast is a totally different thing. I mean, a lot of people love it, but.
You said, I believe, that it had been approved for use in animal feed in North Carolina. If that's correct, what's the process of getting it approved in other states, or is there any national approval process for animal feed?
Yeah, to get it approved in other states, It's much more difficult. You have to go through, almost go through the FDA, which was more paperwork and it was beyond our skill set. So North Carolina, we had tried it. We had data that, well, we had done studies in North Carolina with cattle. We'd done studies with chickens and turkeys and shown that, and had data to show that it was accepted by the animals. So for sort of a provisional use, that was okay, but trying to get a broader claim was, I mean, we'll be working on that, and if we can get a feed company partner, that's where we'd go. But yeah, that's an important point.
What incentive was used to get the cow to put its head into the breathalyzer box?
Yes, so they had a couple of those at Beltsville, and I went out and actually saw them, because at the time we were wondering, well, maybe they would loan us to them, but they don't travel very well. They're kind of big and bulky, and each one is sort of handcrafted, you know, and so, but she said some cows didn't have any trouble with it, you know. You put it in something sweet, and maybe in a couple of days, they'd have their head in it for a few, you know, maybe half an hour or so, which you need to have a period of time. Others took a month. So, you know, it's sort of like, it's animal behavior. Some cats behave one way, others, and you know, we know more about cats, most of us do, than we do about cows. But she said, yes, training could be challenging.
I know you touched on this, but what I'm just wondering, the whole cycle that you're explaining, will it be profitable without some kind of subsidy? I mean, can you really make this thing create a bottom line that makes it a profitable thing? And could you talk a little bit more about that, please?
Well, yes, that gets into some technical details, but the problem is the material that comes out of the brewing process, when you mix everything together, it's like 85% water, so you have to get rid of the water. So that takes quite a bit of energy. And then you have to package it well. So anyone that's worked with hops, they know if they don't keep their hops in the freezer or the refrigerator, they go bad pretty easily. So the beta acids in particular, which have a lot of the strong antibiotic activity, they have a lot of double bonds in them, and they will easily oxidize. So we're working on, we store them in vacuum sealed bags, aluminum bags, so that, you know, that adds an expense. So, so a lot depends on what the application is. If you're trying to feed baby pigs that are under stress, but are very high value, that might be a possible market. If we got carbon credits, and we could show that it lowered methane, and farmers would get reimbursed. But you know, the problem is selling to the agricultural community, margins are very low. So that's what we're working on.
Rusty.
Yes, Nancy.
What was the catalyst that sparked your interest in chemistry?
Ooh. I didn't like chemistry in high school. And I don't know, maybe I like biology much better. But it was the chemistry lab, and I didn't like freshman chemistry either, because I was competing with all these guys from Long Island, or I was an upstater country kid from New York. But it was, when I did a distillation in the sophomore chemistry lab, I ended up wrapping the distillation column with cotton, and I got the best yield. And I said, you know, I observed something and made something happen. So that's kind of, yeah. When I went to grad school, I thought I'd become a polymer chemist because the College of Forestry was very big in polymer chemistry, wood chemistry and making plastics. So not every undergraduate has that background. But then the professor I was going to work with was very strong on thermodynamics and would fill the blackboard 3 times over with equations, and that was not for me. So I joined a biochem lab and worked on yeast and fungi.
How much potential research is being done with regard, that's me,
What?
it's me.
Oh.
How much potential research is being done with regard to the impact on bone health? Because the drugs that are out there
Yes.
have problems.
The problem with, so if you're looking at right now in vivo effects with people, if you will, it seems that hops have a sedative effect, and the Germans have all sorts of natural products, you know, for sleep aid and this or that. They're very big on that, and there's a plant valerian which has some sedative effects. They mix valerians and hops, and there's a small molecule that gets to the brain from the hops. It's a fragment. The problem is that the hop molecules are kind of chopped up in the gut. So we work in the gut because at least they have a chance of surviving and having their antibiotic activity. But the amount that goes into the bloodstream and then gets to the brain or the bone at this point is pretty low now. And the problem is that they're fairly complicated molecules. And my experience working with a drug company is that chemists like to work on simple molecules and not elaborate natural products. So things with, you know, lots of carbons in it, they sort of shy away from. Not that they're not clever and couldn't, but.
You mentioned chlorophyll, and my mind doesn't work the way a lot of people do. If you combine chlorophyll with methane, would that mean that methane has a pleasant smell?
Methane I think is odorless, so it's a, you know, they put some sulfur compounds in it so that we can detect the leak, but yeah.
What do you think what does your team think that is actually happening chemically in the cow's stomach when you add the thyme? Is it the antibiotic effect? Is it binding?
Yes, that's a good question. We're not sure whether it's a direct effect on the methanogens. Those are different than bacteria. They're called an archaea. They have a different cell wall. But the whole process, to make methane, you have to have a generation of hydrogen nearby. So at this point, we don't know. The people that have made Bovaer, or this compound 3-nop, as it's called, they know precisely which enzyme in the pathway it hits. We think it's the hop acids, but we don't know which one, and we don't know exactly where it's working. We had one student who added more hydrogen. We thought, well, you know, maybe it's got something to do with hydrogen. She put more hydrogen in the bottle. And the methane, the thyme still inhibited methane, but then we had another inhibitor, another antibiotic, and that didn't work. So that was not inhibited. So there was something going on, but we don't know the mechanism yet.
Those products seem like they're all organic, coming from the brewing process. Why couldn't they be used agriculturally on farms to improve their soil? Because organic matter in soil is always a big problem, and can't some of them be used in that way?
Well, some breweries have their spent yeast hauled away by farmers, and they just put it in the manure spreader, if you will, and put it on the fields. I mean, it's 40 to 50% protein, so there's a lot of nitrogen in it. And I've thought about using it in compost, but we didn't get to that sort of work. But yes, I think some people haul it away, and I've heard that people that are with sod farms, like to spread the yeast on, so the spent yeast. So it is used in some cases. But none of those provide the brewers with any feedback or income, if you will, from that.
Rusty, maybe you answered this already. If the cow generates hydrogen and has to get rid of it by forming methane, and you stick your thyme in, how's it getting rid of the hydrogen? Is it forming some other compound besides methane?
Yes, yes. So that's one of the problems when they add this compound Bovier. One of the arguments for lowering the methane, besides the environment, is that 10% of the calories of the cow, or the feed that the cow eats, is expired as methane. So if you stop that, then they might grow 10% more, but they didn't see that. So we don't know yet. Sorry to be unclear on that, but yes. And in fact, the problem with getting rid of the methane cycle, if you will, with let's say an anti-methane probiotic, is if they generate all this hydrogen, you have to get rid of it because it is a feedback inhibitor of, digestion, if you will. So it's complicated. People have thought about there are other ways to get rid of the hydrogen. You can convert it into propionic acid, which has a lot of hydrogen on it. So, and that could be good, but that's the metabolism in the gut is, as you might imagine, quite complicated, and it's still not sorted out. And of course, you've got 1,000 different organisms, and they're all interacting and talking to each other.
Any more questions?